First, I'd like to thank the organizers. It's a tremendous pleasure to be here today and honoring Tom. My lab works on two main families of viruses: the filoviruses, these long filamentous particles like Ebola, and the arenaviruses, smaller round viruses like Lassa. They both have RNA genomes that are quite simple. The filoviruses encode only 7 genes. The arenaviruses only 4. It's really quite remarkable that you can achieve such devastating pathogenesis using only 4 proteins. Now, whereas the filoviruses have been associated with one really bad outbreak of 28,000 people, the arenaviruses cause a tremendous global health burden every single year, with hundreds of thousands of cases in West Africa alone, and other viruses in other places. The arenaviruses, the family of viruses to which Lassa belongs, exist throughout the world, on every populated continent. In the New World family of arenaviruses, there is Junin virus, which causes Argentinian hemorrhagic fever. There's Machupo virus, which causes Bolivian hemorrhagic fever. There's Guanarito virus, which causes Venezuelan hemorrhagic fever. Sabia virus in Brazil. Whitewater arroyo virus, which is throughout the American Southwest and causes lethal cases in California. It doesn't call California hemorrhagic fever. In the Old World family of arenaviruses, there's Lujo, which broke out just a few years ago, was found in one Saphire worker who infected 4 people who cared for her, and 4 of the 5 people died. There's Lassa virus, which is the biggie. There's at least 300,000, maybe 500,000 cases of Lassa virus every year. And throughout North America, Europe, and other populated continents, there's lymphocytic choriomeningitis virus, LCMV. Now, LCMV has been used as an immunological model for decades. It also causes a neurological disease, birth defects, and mental retardation in the fetus. 2 to 5% of us are seroprevalent for it. The arenaviruses have just one protein they put on their surface, a glycoprotein called GP. And this is all they have to mediate attachment to a new host cell and driving the virus in. So GP is the target for antibodies and vaccines. And the protein is organized like this. At the N-terminus, there's a stable signal peptide. This sends the protein up to the surface, and it's cleaved from the rest of it in processing. There's a GP1 subunit that does the job of receptor binding, a GP2 subunit that does the job of fusing the viral membrane with the host cell membrane to drive the virus in, a single pass transmembrane domain, and a short cytoplasmic domain. The GP1, GP2 complex together makes a trimer on the viral surface. And it's in a metastable conformation on the viral surface that we call pre-fusion. Upon exposure to receptor in low pH, it will spring into a much more stable 6-helix bundle by the GP2 subunit, with the GP1 subunits released. This is called the post-fusion structure. Over the last decade or so, we've made a lot of progress in structural biology of the arenaviruses. We understand the post-fusion structure of GP2 for Guanarito, California Academy of Sciences viruses, and LCMV. We understand the cytoplasmic domain of Junin virus. And we've had a look at the receptor binding subunits from Machupo, Junin, and Lassa. These are from Steve Harrison's lab and Ron Diskin's lab. And recently, 14-angstrom tomography looking at the conformation of that assembled spike on the viral surface on the bottom right. And so this is what we've been able to visualize to understand how these viruses drive themselves in. What has been missing is a high-resolution structure of the assembled GP trimer that we could use as a template for designing vaccines when we try to understand how the virus gets itself in. And that was the problem we set out to tackle 10 years ago. And in that 10 years, while we focused on that trimer, we got scooped on the other little parts. But we finally got one. And the one we got belongs to Lassa. And the reason we got it was because we worked for a long time with scientists and doctors in Africa to raise the human antibodies we needed to form the crystal contacts, and with the researchers at Tulane that established the public health program there. So this is Sierra Leone. This is one of the countries where Lassa virus is endemic. If we zoom in, this is the capital. This is where you fly into. We drive a few hours to Kenema. And we go to Kenema, Sierra Leone. Because in Kenema is the Kenema Government Hospital. This is the largest hospital for miles around, for hours. At Kenema Government Hospital is the Lassa ward. This is the only hospital ward in the world dedicated to treating Lassa virus. And until the Ebola outbreak, this was the only hospital ward in the world dedicated to treating viral hemorrhagic fever. A lot of infectious disease docs have cut their teeth there. It's hallowed ground for virology. On the campus of this hospital is a biosafety level 3. And that's where we go to work. Now, Lassa is actually a BSL-4 virus. The best we can do in Africa is BSL-3. It's good enough to be careful. I've only stuck myself once. This is now looking into the Lassa ward on the short one on the right, and looking into the Lassa lab. And so Tulane University, this is a program reestablished since the Sierra Leone Civil War, led by Robert Gary, has built a beautiful scientific infrastructure that's extremely collaborative, where anybody from around the world can come and work there. And you want to work there because of the access to patient-derived samples. So, leaving the lab, you can drive a couple hours and through fantastic roads to go out to the villages where Lassa is. This is Dr. Lena Moses of Tulane Public Health. She's been there long enough that she speaks the local language in Mende. And so what the large collaborative group, everything from genetics to structural biology to diagnostics to patient care does, they go to the villages that have been Lassa high. You have to seek approval of the chief to come in and do the work in the village. This is the chief and one of his wives in this picture. And then in the village, there is an ecology team that's part of the collaboration. And these are Sierra Leonean scientists. And what they're doing here is looking at the houses. So the houses are built of mud and sticks. And you can see some tiny little black holes at the bottom. That's where the rodents burrow into the houses. And it's the rodents that spread the disease when they get into food or make aerosols inside the houses. So they search for evidence of the rodent infestation. They set the traps. They sero-survey the rodents in the village. They consent and sero-survey the people to try to understand how Lassa virus is spreading, or their agricultural practices that drive the virus into the village, or the food storage practices that prevent people from being contaminated. And then as night falls, the ecology team gathers the villages, hangs up a bed sheet, runs a generator, and shows a movie off a laptop that educates the people about Lassa virus and how to avoid it. And then there's a treatment program for free where the Kenema Government Hospital will come and get them in Orlando and give them free treatment. On one of our trips, one of the villages was so grateful that we had come there to work in Lassa because they had lost a lot of members, so they gave us a goat. And they made me hold the goat because the structural biologist isn't used for much else at that point. We did go get lunch and run a Eliza and hold the goat. But where we are useful is in engineering the glycoprotein. And so for 10 years, we worked in this circle where in La Jolla, my graduate student, then postdoc, now staff scientist through the years, Kate Hasty, and her technician, Michelle Zincanati, steadily engineered the glycoprotein until it was a homogeneous sample that behaved well for structural biology. We worked with Bob Gary and James Robinson in Tulane, and Dr. Sean, Dr. Grant, and Augustine Gova, and all of their staff in Sierra Leone to engineer glycoproteins, use the glycoproteins to identify human antibodies and survivors, use those antibodies to go back and make better glycoproteins, and ultimately try to get a structure at the end. So from the staff in Kenema, we did identify human survivor antibodies. Now all of the staff, whether they work in the clinic or in the lab, or they drive the ambulance that goes and gets the patients, they got their jobs because they're Lassa survivors. So they're immune, and presumably there's some level of occupational exposure. They had screaming high antibody titers and were able to identify some fantastic antibodies from cells donated by the people. And this is what we found from that study. The very best antibodies, those that neutralize the virus most potently, are specific to the assembled trimer. They bind to quaternary epitopes. They aren't directed against the post-fusion GP2 or the disassembled parts. From those antibodies, we were able to crystallize the Lassa GP trimer, and that looks like this. This is one of the human survivor antibodies from Sierra Leone, found in the Lassa glycoprotein trimer. Now let me go to Primal, and I'll show you what it looks like. So here's a ribbon view of the glycoprotein trimer. There are 3 monomers here, purple, green, and orange. I've colored the receptor binding subunit in a lighter shade and the fusion subunit in a darker shade. A little hard to tell on the screen. But the receptor binding GP1 is like a ball scoop of ice cream. And it sits inside a cone made of the GP2, the assembled helices that together make kind of a bowl shape where the ice cream scoop sits in. And then the receptor binding GP1 has a long melted drip running outside that ice cream cone that makes a beta strand to anchor it together. GP1 at top, GP2 at bottom. If you roll over the trimer and you look at the center, I'll show you where it's glycosylated. Each monomer bears 11 potential end-link glycosylation sites. And we see every one of those glycans in the crystal structure. So most of the molecule is cloaked with glycan. There are only a few sites that are left exposed. And this is where the human antibody binds, at the base in one of those exposed sites. And you can see actually what it does, and the reason why it's specific for fully assembled trimer is that it binds in between two monomers. So each antibody fragment simultaneously binds half of one and half of the other. Now we actually saw two structures: a potently neutralizing antibody in gray and a weakly neutralizing antibody in red and pink. Now what I've done here is I've recolored the parts of the fusion subunit. I'll show you what they are. Here they are. The first heptad repeat is going to become one long single helix in post-fusion. Here you can see how it's broken into multiple different segments. It's circled receptor binding subunit. The second heptad repeat is green. The T-loop in between is hot pink. And the fusion machinery is light blue. Now some viruses have a fusion peptide, which is a pre-N-terminus. Some viruses have an internal fusion loop. It looks like Lassa virus uses both. So that's the fusion structure. And the antibodies recognize a lot of different parts of this fusion machinery. The best we can figure out why the potent antibody is potent and the weak antibody is weak are the number of contacts and the affinity of the antibodies. As the potent antibody makes a lot of contacts with a higher affinity than the weakly neutralizing antibody shown here. Now if we roll back again to look at the top of this trimer, and we can map the amino acids that we know are essential for binding its first receptor. Now Lassa virus has at least two receptors. There's an extracellular receptor where it binds a sugar on the surface of alpha-dystroglycan, and it has an intracellular receptor. Once it gets into the endosome, it binds LAMP1. There were other molecules involved as well because the virus will happily also infect alpha-dystroglycan knockout cells. But the residues that we found that are essential, we and Michael Oldstone over many years, are essential for binding alpha-dystroglycan map here. They map to the trimer interfaces. I'm showing you just the ones from the orange one. And so what we think this is telling us is that alpha-dystroglycan also needs an assembled trimer to bind Lassa. And indeed, the receptor binding subunit expressed by itself as a monomer does not bind the alpha-dystroglycan receptor. The intracellular receptor, LAMP1, requires a histidine triad that was identified by Ron Diskey in Israel. And the histidine triad is on the upper and outer surfaces in that beta saddle. That's very similar to the place where the South American hemorrhagic fever arenaviruses bind their receptor, transferrin receptor 1. Okay, so if we color again GP2 by its parts, that's what GP2 looks like in its pre-fusion conformation. This is what GP2 looks like in its post-fusion conformation. That's the monomer. Here's the trimer that makes the 6-helix bundle. So you can see the broken yellow helical segments will rearrange into a single long rod at center. The green peptide repeat 2 will collapse up, bringing the viral membrane and fusing it with the host cell membrane. And there's a conformational change in that hot pink T-loop from a strand to a helical structure. And the fusion machinery is invisible with post-fusion structure, but it's presumably up in the host cell membrane. So back to the trimer. Okay, and we think we've got the right thing because it fits pretty nicely into that tomography from he's been in his lab at Oxford University that he was Thomas Strecker at Marburg. And so we think what we've captured in the crystals here, bound by that human antibody, is a similar conformation to what's on the viral surface. Now that's the first trimer that we've seen at moderate to high resolution for the arenaviruses. So we'd like to try to use it as a model to understand what we can about all the other disease-causing arenaviruses as well. So this is Machupo GP1. solved by Tom Bowden and Jonathan Abraham, complex with its transferrin receptor 1. If we align it with the receptor binding subunit of Lassa, you can see that the beta sheet core is similar, the loops on the surface are different, but it's similar enough, and the GP2s are similar enough in sequence, that we can try to model Machupo onto the GP trimer. And because the structure of Machupo was solved in Steve Harrison's lab, which was transferrin receptor 1, we can model what that might look like with the receptor binding to the New World arenaviruses. Now, 3 will fit in this model, but of course, transferrin receptor 1 is actually a dimer, so we don't know that 3 bind in the actual trimer. We need to do that experiment. That might be what at least one of them would look like. Hunine virus is a little bit different. It bears fewer glycans. It has a single critical extracellular receptor, and you can generate neutralizing antibodies just against GP1. And this is one of those. This is another structure dissolved by Steve Harrison and Jonathan Abraham of an immunotherapeutic against the Argentinian hemorrhagic fever virus bound into the receptor binding GP1. And so if we model that in the trimer, we can see what that might look like if 3 antibodies are able to bind at the same time. Now here's probably the most interesting thing. What we solved was Lassa at pH 8, anchored in the pre-fusion trimer. Just about a year or 2 ago, Ron Diskin's lab in Israel solved the Lassa GP1 by itself at pH 5, and that's this structure here. If we overlay the GP1 at pH 5 with the GP complex at pH 8, you can see that beta saddle in the center is conserved, but there's a lot of other differences. For example, what I'm showing you in brown arrows, the broken helices alpha 1 and alpha 2, instead form one long single helix, and GP1 is by itself at low pH. This structure, which is a helix and a loopy bit in the assembled trimer at basic pH, is instead a beta sheet when the GP1 is separated from GP2 at low pH. And the termini are moved. The GP1 in isolation doesn't have its termini anchored to the receptor binding subunit, and they've moved upward. Whereas in the assembled trimer, one is continuing in its polypeptide at GP2, and the other one is buried down in the trimer nucleus. And so if you're going to morph one PDB from the other, this is just a stupid morph. We don't have biological evidence to support this yet. But just comparing the one crystal structure to the other, if you start at pH 8 with the GP1 and complex, and you finish at pH 5 with GP1 alone, you can see the separate helical segments here. These termini interact with GP2. Here's a helical term. If you morph it to the other PDB, you can see it rearranged. The beta saddle in the center stays relatively constant, but the helices around undergo significant conformational change. And this could be important because, as a virologist, you learn on your mother's knee about conformational changes in the fusion subunit, where it collapses from its pre-fusion to its post-fusion structure. But there's possible evidence here for conformational change in the receptor binding subunit as well. That could be what pries it off as GP2 as the process goes. So it's made a single helix, it's formed as beta strands, and its termini are moved away from GP2. This is how we think the antibody might work. Now, the work from Oxford and the tomography showed us the compact globule of the spike on an actual loss of variant. And they also looked at virus-like particles treated with low pH in the LAMP1 receptor. And they saw that the structure was balloon open. The light blue density here belongs to LAMP1. Now, LAMP1 is bigger than that, so maybe this is telling us that it wasn't a complete occupancy, or maybe it was flexible. But certainly that immunogenesis suggests that LAMP1 binds at the top. This antibody FAB fragment binds at the bottom. It doesn't, certainly an IgG could sterically interfere, but the FAB itself doesn't immediately look like it would. Yet in the experiment at the top, we can see that in the presence of the FAB fragment, the gray line, LAMP1 binding is hindered. And so it could be that because this antibody is quaternary in its epitope, it anchors that glycoprotein together and prevents it from moving open in the way that it needs to bind the LAMP1 receptor at low pH inside the endosome. Now, what we've learned for Lassa and studying the antibodies from human survivors are that the best antibodies bind the quaternary epitopes, only the assembled, stable, pre-fusion trimer. They don't bind the disassembled parts, whether it's an isolated GP1, a post-fusion GP2, or some trimer in the process of disassembly. And this is not a new story. I mean, this is the HIV story, as Ian will tell you, and we certainly learned a lot by being in Scripps. But here's the trouble. The natural glycoprotein for Lassa virus makes all of these. It is not a stable protein. It took us a long time to figure out how to anchor it in one place. If the natural glycoprotein makes all of those, they're all exposed to the immune system, and they elicit a variety of non-neutralizing or poorly neutralizing antibodies. And if you put your natural GPC on the surface of a vaccine particle, you will also get that variety of structures. And indeed, there is no vaccine for Lassa. There is no treatment. All of the candidate vaccines that have been tested have arrived at the conclusion that what correlates with protection in the animal model is T cells. T cells recognize chopped up peptides. They don't care if the protein is folded properly or not. The Lassa vaccine dogma is that antibodies do not correlate with protection. And many of the Lassa vaccine makers are not even seeking an antibody response or focusing on trying to figure out how to maximize the T cell response. But here's what we think. Now that we know what this looks like and how to make it, couldn't a stable engineered trimer help? If we could present this on our vaccine platform, we may be able to elicit both neutralizing antibody and T cells, and surely if we pair them together could offer better protection than T cells alone. Now, we don't know for sure, but we'd sure like to try. And so we're hoping that this structure, which was a long time coming, can serve as the blueprints to design a much-needed Lassa virus vaccine. Now, all of the credit for this structure goes to Kate Hasty, who stuck with this for 10 years. She came to my lab as a graduate student in 2007, and in her first committee meeting, she told her committee, I'm going to solve the arenavirus glycoprotein. And they looked at her and they said, good luck, lady. And she stuck with it until we got it to work 10 years later. She could solve a lot of structures along the way, so she could get her PhD and get her postdoc. But the credit goes for her for handling it. Also her technician, Michelle Zandonati, that screened hundreds of constructs for her, binding many, many antibodies along the way. So here's Kate in La Jolla and working in Africa. We'd also very much like to thank Bob Gary and Jim Robinson at Tulane University, who have built this collaborative Lassa fever program in Kenema and raised all those antibodies and did the analysis to help us find the ones that we needed to grow the crystals. But also like to thank the staff in Africa themselves, Dr. Graham, who sees the patients and helps us obtain the antibodies, Augustine Gova, who runs the VSL3, Mambo Momo, one of the technicians in the lab, Samira Jala, the program coordinator, and Louis Branco at Zalchek, who's also instrumental in moving his antibodies forward in animal models. One of my favorite African proverbs is this: if you want to go fast, go alone. If you want to go far, go together. And along the lines of going far together, we're hiring. After a couple years of fundraising, we just bought a pretty nice cryo. And so if you have somebody in your lab who's finishing up, who wants to work on really interesting projects and live in San Diego and have a PI who adores them, please send them my lab. And I'm happy to answer any questions.
Questions? We have lots of time for questions, and we have to give them time.
So you were saying that the way the neutralizing antibodies are binding is not immediate why they would be stopping the interaction with the receptor. But I was wondering, so I thought that what they were doing is preventing the transition to fusion, right? Because they're binding at a site that is going to be completely transformed. So do you need to evoke something that has to do with the receptor binding to explain how they work?
Absolutely. They are definitely preventing fusion. There isn't a good fusion assay in the field, but the best we can find is that they prevent those low pH conformation changes, and that's a primary mechanism. But just for jollies, we wanted to see if they had an effect on lamp-like binding, and we didn't expect that they would, but they do diminish it a bit. And so based
talk and beautiful work. Thanks. The conformational changes that happen in virus development are just been amazing for the last, looking at them over the last 30 years or so. And I'm wondering, what is the status of modeling and simulation of these kind of changes, in particular for Lassa, but in general? Can you just sort of summarize the state of that field?
The state of modeling
Of the conformational changes that are going on.
Ooh, we need better work in that area. Kelly Lee has done some work on that for influenza. Bob Lamb has looked at this for parainfluenza viruses and others. But much of what happens there is still a black box because it is technically difficult to capture what's in the middle. There are investigators that are now starting to try to capture those intermediates by arresting them with different peptide inhibitors or by using the advances in cryo-EM. But there's definitely a need to try to understand that better because it's a clear therapeutic target. And the other thing we need to understand is how many of those events do we need in the same place for fusion to happen? I mean, the assumption is 3 might work to make a pore that might be enough to rip it in. But if you look at the other viruses, look at Ebola virus, it's a micron long. Must the whole thing fuse, or is a small cluster of a couple of glycoproteins in one area enough to allow the genetic material in? Nobody else.
I mean, I just had a question. You left us with kind of a cliffhanger. What sort of strategies or guesses do you have for stabilizing and making the vaccine?
We followed a strategy that was much like the SOSIP strategy for HIV. So we hunted blindly for years looking for disulfides we could engineer in that would anchor GP1 to GP2. So part of the problem is that GP1 and GP2 must be enzymatically processed from each other in order to make a functional spike. And it turns out we see that that's because the termini need to translate 30 angstroms from each other in the trimer from the uncleaved structure we saw for LCMV. And so we had to have cleavage occur, and GP1 is likely to float away from GP2 in the spring GP2. So we had to hunt around to find a disulfide that would anchor them together and still be properly folded protein. And then we had to engineer prolines elsewhere, and there was one more hydrophobic mutation we had to engineer in to sort of stabilize the GP2s together.
Do the rodents form antibodies?
No one has gone and bled them, and that's a really interesting question, and I would like to know. They don't get sick, but they carry it, so presumably they would. And the other thing I'd like to know are, there's when virologists rode around on horseback and they figured out Machupo virus outbreak in Bolivia, they discovered that houses that had cats didn't get Machupo. And so the cats are eating the rodents. How come the cats don't get sick? I don't know. I would like to know. No one's looked much at the rodent antibodies.
Over here, blood.
Fantastic talk. Thank you. I think that the take-home lesson, especially for young people, is your slide, the one before last, collaborate. Yes, this is an extremely important message. And without all these things which you have done in Africa, it wouldn't be possible. Yes.
It's true, we only got the crystals because we had the antibodies. We only got the antibodies because of people willing to donate them, and the people willing to build the facility we needed, and the people willing to work in that hospital and that lab.
I have a quick question. So the conformational change from page 5 to 8, where you have the straight helix and then straight dots from the specimen for the pharmacologist. So if you look at the trimer, I mean, this really the broken helix looks energetically very unhappy. Can you sort of say whether this is basically energetically failed over the course of interactions within the trimer, or is it really just the pH?
It is quite unstable. Without stapling it together, it really wants to fall apart. Yeah. And so whatever interactions there are in the trimer are only barely just enough. And springing into that post-fusion structure is energetically favorable.
Let's thank Kevin again for just a very inspiring